Hello everyone, and thank you for listening to this presentation. Today I'll be presenting the topic about the significance of arbuscular mycorrhizal fungi and its association with the plant diversity. Two research papers are included in the project. One was conducted by Ganj and his colleagues in 1993, and the other study by Vogelsang and his colleagues in 2006. These research papers examined the effect of mycorrhizal fungi on the diversity, species richness, and productivity of plants. On the left side, figure 1 shows a plant whose root was not colonized by mycorrhizal fungi, while on the right side, a plant whose root was colonized by fungi. Before we jump to the two articles and find what the authors were trying to investigate, it is important to know what is mycorrhizal fungi, so we can form a basis of understanding for the topic. Mycorrhizal fungi are an important member of plant microbiome that improve plant access to water and nutrients. Ectomycorrhizal fungi and arbuscular mycorrhizal fungi are two dominant types of mycorrhizal fungi and form a symbiosis with a large number of terrestrial plants. In addition to that, all plants synthesize sugars through a process of photosynthesis, and sugars that represent a source of energy move to different parts of the plant that includes root. A crucial factor for the survival of fungi that is found at the root of plants is sugars such as the glucose and sucrose, and plants provide a constant supply for these sugars to fungi, thus allowing them to survive at the root. However, fungi infect plants to achieve better absorption of water and nutrients from the soil. So, mycorrhizal fungi found on the roots of plants create hyphae that improve the absorption of plants. Although absorption is a major function of mycorrhizal fungi, they also improve the presence of phosphorus and nitrogen, increase plant resistance to soil microbes, plant species, and resistance to adverse conditions such as drought. In 1998, the first study was conducted to examine the effect of colonization of microbial fungi on plant diversity, and many more studies have been conducted since then. And the complex interactions that take place between different plant species and mycorrhizal fungi that colonize their roots determine the functionality and stability of terrestrial ecosystems. Arbuscular mycorrhizal fungi allow plants to grow under stress conditions such as drought, temperature, high soil salinity, and high metal concentrations in the soil. Figure 3 here shows the large diversity of plants in the low density area. So mycorrhizal fungi have a role to play in such a large diversity in a small area. And fungi allow plants to grow in soil where the nutrients content is low through a complex interactions with the plant root. So it is important to examine the colonization of roots by mycorrhizal fungi because they can greatly contribute to plant diversity. Ganj et al. 1998 treated the soil in Silwood Park in the United Kingdom with fungicides to determine the effect of mycorrhizal fungi on plant diversity. As control soil, they used plots that were not treated with fungicides and contained natural amounts of fungicides. After fungicide treatment, the authors monitored plant growth for 2 years and performed sampling. The fungicide treatments were performed late in March, and in the period from April to October 1988, the authors collected vegetation samples for periods of 3 weeks, followed by another 3 years at monthly intervals during each growing season. In 1990, the authors collected plant samples at intervals of 3 weeks over a year, then at monthly intervals. And to determine the influence of mycorrhizal fungi on the number of plant species, the authors determined the number of plant species growing on the treated soil and compared it with the number of plant species grown on untreated soil. The soil was bare before the start of the experiment, and the authors wanted to find out how the lack of fungi in the soil affects different types of plants. Figure 4 here shows plants with roots. So on the right side are plants that grow in the presence of mycorrhizal fungi, and it can be seen that the root is much more branched compared to the root of plants on the left side of the picture, where mycorrhizal fungi were not present. Vogelsang et al. 2006 planted potted plants in which soil was sterilized without mycorrhizal fungi, soil treated with one species of mycorrhizal fungi, and soil treated with 6 species of mycorrhizal fungi. Sterile soil was used as a control, and the authors used perennial high-grass prairie plants, which are plants that have a lifespan of several years. Also, the authors added various sources of phosphorus to the soil in which the plants grew, and after the addition, when mycorrhizal fungi were present in the soil, there was an increase in the biomass and plant diversity compared to the values obtained for plants that grew in soil in which there were no fungi present. 23 weeks after the treatments, the authors collected and numbered the plants by species. Figure 5 here on the left shows plants in which soil containing mycorrhizal fungi, and on the right side, plants in soil without fungi. The plants on the right side of the figure are larger and more lush compared to plants that grow in soil in which mycorrhizal fungi are not present. In these two images, we can see the squares used to sample the plants in the field. So plants that are within a square are collected and their species is determined. Also, their mass is counted and measured. Figure 7 here shows the accumulated number of plant species in fungicide-treated soil and fungicide-free soil. The triangles in the figure represent the accumulated number of plant species that grow in the fungicide-treated soil, and the squares represent the accumulated number of plant species that grow in the fungicide-free soil. The y-axis shows the cumulative number of plants, while the x-axis shows the year of plant sampling. Figure 7a shows the cumulative number of plant species for the period of 1988 to 1991. At the beginning of the 1988 experiment, the number of plant species was the same at treated and untreated sites. However, during 1988, there was an increase in the number of plant species in the soil that was not treated with fungicides compared to the number of plant species that grow in the soil that was treated with fungicides. After that, and by the end of the experiment, each year the number of plant species was higher when the plants grow in fungicide-free soil compared to the number of plants grow in fungicide-treated soil, as can be seen from figure 7a. Figure 7b shows the number of plant species that grow in the fungicide-treated soil and were not treated between 1990 and 1991. Also, at the beginning of the experiment, the number of species was the same at both test sites, and over time, there has been an increase in the number of plant species in non-fungicide treated sites compared to the sites where the soil has been treated with the fungicides. Figure 8 shows the plant community productivity and diversity response to arbuscular mycorrhizal fungal richness. The logarithm of plant biomass is shown in A, and plant diversity as Shannon Diversity Index in B are both shown on the y-axis. The x-axis shows the number of species of mycorrhizal fungi with which the soil was treated in A and B. From figure 8a, it can be seen that the richness of mycorrhizal fungal species affected plant biomass. So when the plants were growing in the soil without mycorrhizal fungi, the plant biomass was the lowest compared to the biomass of plants from the soil with fungi. Although there was an increase in the plant biomass when the soil was treated with one species of fungus alone, there was no large increase in biomass from approximately 2.75 biomass increased to approximately 3. And when the soil was treated with 6 species of fungi, the plant's biomass was the largest compared to other treatments, and it was approximately 3.57. Figure 8b shows the diversity of plants depending on the richness of mycorrhizal species. So the lowest plant diversity was when the soil in which the plants grew was not treated with mycorrhizal fungi. Then when it was treated with one species of mycorrhizal fungi, and when the soil was treated with all six species of mycorrhizal fungi, the diversity of the plants was the highest. Shannon Diversity Index was approximately 0.7 for plants grown in soil that was not treated with mycorrhizal fungi, and for plants grown in soil treated with one species of mycorrhizal fungi, it was 0.98. And for plants grown in soil treated with six species of mycorrhizal fungi, the Shannon index was approximately 1.09. The results of the experiment conducted by Ganj et al. 1998 lead to the conclusion that mycorrhizal fungi improve plant growth. So when no mycorrhizal fungi are present in the soil, fewer cumulative plant species are grown compared to plant species that grow in soil where mycorrhizal fungi are present. Also, the use of fungicide treatments kills mycorrhizal fungi, which leads to reduced plant diversity. Based on the results of Vogelsang et al.'s 2006 research, it can be concluded that the richness of mycorrhizal fungal species affects plant productivity and diversity. So when more species of mycorrhizal fungi are present in the soil, there is a greater diversity of plants compared to the soil without mycorrhizal fungi. Also, arbuscular mycorrhizal fungi, or AMFs, live in symbiotic communities with the root of approximately 80% of terrestrial plants, and the symbiosis between AMFs and the plants can be antagonistic to each other, and this depends on many factors such as plant type, type of interaction, and available nutrients in the soil. Mycorrhizal fungi create a connection between the soil around them and the roots of the plant. This allows fungi to take nutrients from the soil such as phosphorus and nitrogen that are necessary for plant growth. So the mycorrhizal fungi increase the surface area of the plant's roots, and thus the plant has a larger surface area for absorbing nutrients from the soil and water and increase plant growth. Figure 9 here shows the positive effects of AMF infection, and the hyphal zone of AMF extends beyond the depletion zone of the plant. The hyphal zone is gray in the figure, and extension of the hyphal zone provides plant with access to a larger area of soil. Also, the depletion zone of phosphate mycorrhizae is formed around hyphae, which is purple zone in the figure. In addition to phosphate, other substances are included, such as zinc and nitrogen, and through the induction of systematically acquired resistance, MF protects plants from harmful stresses, such as increased soil salinity, drought, pathogens, etc. As I explained in the previous slide, fungicides are used to kill fungi that cause diseases in plants, but fungicide treatments also kill mycorrhizal fungi. Therefore, it is necessary to evaluate the results of fungicide treatment before applying the treatment. Also, before using fungicides, it is necessary to establish the correct diagnosis of plants and apply as few concentrations of fungicides as possible. Thank you for listening, and here is a list of references that I have used for this presentation. I would be very happy to answer any questions that you might have about the data presented or the results, and thank you again.